Good morning, everyone, and welcome to the second part of this cycle of conferences in which, under the title of Chemistry, an Outreach to Society, 11 talks related to chemistry, chemical engineering, and biochemistry will take place. Today, in this second Thursday of this cycle, it is a pleasure to me to introduce to Elena Benito Peña from the Department of Analytical Chemistry, who will speak about an attractive topic about bioinspired materials for optical sensing. As we usually mention when we introduce these talks, we have chosen the online format instead of face-to-face, taking advantage of the platform provided by the General Foundation of the UCM, to whom I want to thank the help and support to organize this activity. We thank also to our finding sources, the Spanish Biochemistry and Molecular Biology Society, the Royal Chemistry Society, Sectorial of Madrid, and Vice Rector of Research and Transfer for their financial support, because this enables us to use this format, and this way many more people can be connected. The Spanish language is extremely rich in terms and expressions, but science is mainly written and discussed in English. Our junior researchers who have been investigating different aspects of the chemistry return to our institution after spending a long period abroad. One of the reasons we organize these webinars in English is because they are very familiar with the use of this language to report the scientific results. In this way, we can broaden our audience to foreign institutions, some of which have been the host institutions for these researchers. The lectures will take place on Thursday at 12:30, and the last one will be June 2nd, and it will be announced at the beginning of the week, including the link of the registration form. Students who have enrolled for credit for this activity, please let me know by emailing me at the address given in the announcement of this activity. Without further ado, I give the turn to the moderator of this talk, Maricruz Moreno Bondi, professor of the Department of Analytical Chemistry, who will introduce the speaker and moderate the questions that you can write in the Preguntas and Respuestas button in the lower side of your screen. I have to say also that the questions have been written in English or in Spanish if you prefer. Then now, Professor Moreno, is your turn.
Thank you, Maite. It's a pleasure for me to introduce you to Elena Benito Peña. Elena received her PhD in Analytical Chemistry at Complutense University of Madrid in 2006 on a pioneering work in Spain on the application of molecularly imprinted polymers for the development of optical sensors. After a postdoctoral stage at Complutense, in 2009, she moved to Tufts University in Boston to work in the group of Professor David Wolf on the development of microarray-based biosensors and single molecule analysis. She was also involved in a very interesting work in collaboration with a group of Professor Whitesides at Harvard University, focused on the application of infobiology, particularly on the implementation of arrays of microorganisms colonies for time and on-demand release of messages. Elena returned to Complutense in 2011, and since then, she has been involved on the search of new biomimetic recognition elements for the development of optical sensors and new analytical separation methods. During the last years, she has especially focused on the application of phage display to the selection of peptides and recombinant antibody fragments, together with the implementation of new signal amplification techniques using molecular engineering and nanoparticles for the sensitive analysis of food contaminants. As Professor Villalba mentioned previously, the title of her presentation is Bioinspired Materials for Optical Sensing. Elena, the camera is yours.
Thank you, Maricruz. Good morning to everyone. Thank you to the organizers, to Maite as well, for giving the opportunity to show you some of our work in this live webinar. Let me share the presentation with you. Okay, so let's see if this works. Yes. So first, let me briefly introduce you my research work. It is my research group. It is the Grupo de Sensores Químico-Ópticos y Fotoquímica Aplicada, GESOLFA. We are in the Faculty of Chemistry at Complutense University of Madrid. And from the many research lines that we are working in, some of them are devoted for the development of optical sensors and biosensors using nature mimicry recognition elements. Biomimicry is a discipline that seeks to find technological solutions inspired by the design of nature. Nowadays, we are quite able to adapt the solutions provided by nature to real-life challenges. For example, in the slide, you can see how this structure, that is a marine sponge, is similar or is inspiring the buildings that are done time ago by Norman Foster at London City. The applications of biomimetic materials are numerous and range from anti-reflecting coatings, diffractant pigments, adhesives, membranes, and what is the topic of this speech today, molecular recognition elements. Most biosensors are based on natural recognition elements, such as antibodies, enzymes, whole cells. Their use offers well-known advantages, for example, high sensitivity and selectivity towards the target analyte. However, they also present some limitations, such as limited stability, difficulty of preparation, or even high cost in the production, that in some contexts it could be difficult to handle with. For all these reasons, we are working for some years ago in replacing these natural receptors by other created artificially, either by chemical synthesis, like molecular imprinted polymer, or synthetic biology, such as phage-derived peptides and antibodies. Also, we can use recombinant proteins with different functionalities for sensing applications. In our research group, we develop sensing platforms on demand. We're using either natural or artificial receptors, but always using optical transductions. Some examples, as you can see in the slide, are based on arrays, like could be either planar or bit-based arrays, fiber optical sensors, that it was of the beginning of the group, magnetic bits assays, also like in the last years is incrementing exponentially is the development of homogeneous assays, especially because of the simplicity of using them. And of course, readout that can be done with the smartphones. I mean, it's true that the cameras that has built in the smartphones nowadays enables the use of these devices for analytical purposes. An interesting application of Bioinspired materials is the development of sensing platforms for the analysis of mycotoxins. Mycotoxins are secondary metabolites produced by filamentous fungi that can cause both acute and chronic diseases. Because of this, European Union has established maximum levels for them in food and feed, although despite the efforts, the occurrence is still growing, causing economical losses. Competitive immunoassay and immunosensors are now widely used for mycotoxin analysis because, comparing to chromatographic methods, they provide results with high sensitivity and selectivity in a simple and inexpensive manner. However, there are problems associated with the manufacture and use of mycotoxin conjugates in terms of, for example, the complexity involved in the preparation and the inherent risk of working with substances that are actually very toxic. So what is the alternative that we offer? The replacement of these toxin derivatives by mimotopes. Mimotopes are peptides that mimic antigen epitopes in the way of binding to the corresponding antibody. To select the mimotope of each toxin, toxin that could be another analyte, but in this case, mycotoxins, we have used a very relevant technique in the field of biotechnology, which is phage display. Phages are extremely robust and stable virus particles, which use bacterial cells as hosts to replicate. Specifically, we have used a library of phages displaying short peptides with different amino acid sequences, what we have called phage-derived peptides. The key point is to find that peptide out of all the billions of possibilities that mimics the toxin in binding to the antibody. For this, we follow a secret procedure known as panning, which is executed in a series of well-defined rounds. First, the phage library is exposed to a surface coated with the antibody selected to the mycotoxin. After an incubation step, we wash out all the phages that did not bind to the antibody. In the third step, we elute those phages that specifically bond to the antibody. Then, the eluted phages are amplified by infecting them with the E. coli cells. So we increment notably the concentration of virions specific to the target antibodies. Finally, the panning is repeated between 3 and 5 times to guarantee a high level of specificity and binding strength. After giving a flavor of how the epitope-mimicking peptides are obtained, I will explain you more specifically how we first identify the mimotopes for, in this particular case, for 3 mycotoxins, fumonisine B1, T2 toxin, and cerealinone, obtained by phage display. And second, I will show you a couple of biosensing platforms that we prepare based on them. Regarding the mimotope selection, after performing those 3-5 panning rounds, we evaluate the specificity of those polyclonal phages that has been selectively eluted. For this, we run ELISAs in which we can see how there is an increment by the rounds of those phages that specifically binds to the antibodies, whereas for the negative controls that are well coated with BSA, they are not incremented in the same year. But that means that we are obtaining peptides, phages that are selective to the antibodies and not to the other proteins that could be there. The clone selection, I mean those clones that are able to compete with the analyte for the specific size of the antibody, these individual variants should be also selected doing a phage ELISA. In this case, a competitive assay was performed between the monoclonal phage and the corresponding mycotoxin for the specific size of the antibody. The way of detect this immunoreaction is using a detection antibody that is an antiphage labeled with HRP. And the signal is obtained enzymatically using a fluorogenic substrate in this case. With this approach, the calibration curves obtained for the 3 mycotoxins are presented in the slide. And as you can see, the sensitivity obtaining for the 3 of them is really nice and is in quite agreement with the legislation requirements for the detection of these contaminants. And also the reproducibility was very nice. Looking at the peptide amino acid sequences, we can compare the frequency of occurrence of amino acids of the original library with the frequency of the amino acids of the clones displaying the mimotope selected. Interestingly, what we can conclude is that in the case of the fumonisine mimotope, the increment in the negatively charged aromatic amino acid is significant, what makes sense regarding the structure of the fumonisine B1, which has 4 carboxylic groups. In the case of the cerealinone and the T2 toxins, the performance is different. In this case, these molecules are more hydrophobic. What that means is that the increment in the more hydrophobic amino acid is obtained. Next step is getting out of the virus and using phage display derived peptides with different modifications. In our particular case, we explored 2 options. The first one is the preparation of synthetic mimotope. And the second is the production of luminescent recombinant mimotopes obtained by synthetic biology. An example of the first case is the synthetic mimotope microarray prepared for the analysis of fumonisine B1. In this case, a biotinylated mimotope, the one of the fumonisine B1, was immobilized in neutropidine spotted slides. The assay format consists in an indirect competitive assay using a detection probe that was an anti-IgG antibody tagged with a fluorophore, that is Alexa-Fluor 647. With this approach, the figure of merits were pretty good, and the analysis of the real samples also show good accuracy, reproducibility, and nice recovery. In all the cases, the quantification was performed in agreement with the legislation that is settled for this mycotoxin. Thinking in the selectivity of the approach, we can see that it was quite interesting and quite nice, because we are not having any cross-reactivity with other mycotoxins, but only with the fumonisine B2. That is pretty similar to the target, and actually the legislation doesn't distinguish between both of them, and it asks for the detection of the sum of both. For the second alternative, the construction of luminescent recombinant mimotopes, in this case, there are several advantages of using molecular biology for tagging the mimotopes. For example, we don't use phages and neither secondary antibodies for the assay development. Also, the production is easy. We use bacteria that is relatively cheap. It's possible to have an ad hoc selection of the fluorescent protein, just thinking in what kind of excitation and emission you require, so you can select from the descent of variety of different fluorescent proteins that are in the market. You can select the one that is more appropriate for your system. And also another advantage is that after cloning, you can have a continuous supply of the fluorescent mimotope. The example that I want to show you with this approach is an homogeneous fluorescent protein-based immunoassay. The working strategy in this platform is basically devoted by the performance of the protein, of the fluorescent proteins, when this binding to the antibody or not. So that means that when there is no analyte, the fluorescent mimotope binds to the antibody that is linked to a gold nanoparticle. That implies that there is a quenching of the fluorescent protein because a surface energy transfer. However, when there is analyte in the solution, the mimotope that is fluorescently fused, labeled, is far away from the gold nanoparticle, and with this big distance, the quenching is avoided, and you can return, and the fluorescence of the fluorescent protein is recovered. The best condition for performing the assay implies a concentration of 1 nanomolar of the gold nanoparticles, because allows a maximum quenching efficiency and also reduce the possibility of non-specific binding. Also, the potential effect of a very strong light absorption and scattering caused by the gold nanoparticles is also minimized. About the cross-reactivity, in the case we can observe the same performance like in the other approach, basically because the selectivity is more devoted for the antibody type that is used. So we don't have any cross-reactivity for other mycotoxin, and only fumonisine B2 is the one that gives signal in the approach. Regarding the analytical characteristics, the limit of detection was even better than the limit of detection obtained with the biotinylated peptide used in the microarray platform. The results with the real samples were also really interesting and nice, and are in agreement with the legislation requirements. The last example I want to show you is the use of optical sensing with a different kind of bioinspiring material, and is molecular imprinted polymers. What is a molecular imprinted polymer? Well, it's a synthetic polymer with cavities of predetermined selectivity that can be tailored to mimic the molecular recognition ability of certain biological species, like could be antibodies, enzymes, and another kind of receptors. In general terms, the step to obtain an imprinted material is easy. First, we have to select the template molecule. That could be the analyte itself or a surrogate, that is a molecule pretty similar to the target one. Second, the template and the functional monomer are mixed together to get an spatial assembly. After the arrangement in solution, this construction is fixed through the polymerization in the presence of a cross-linker and an initiator of that polymerization. And finally, what we have to do is take out the template to have the cavity that is going to specifically bind the template molecule and those the analyte of interest. That means that if we do a rebinding of a molecule that is not similar to the target one, it's not going to bound the polymer. But if we do a rebinding with the analyte that is of interest, we can get the perfect match. Some of the advantages that we can observe in molecular imprinted polymers for being used in sensing is the high stability. They are really stable polymers. They are tailor-made materials, so in principle, any compound can be imprinted for these purposes. They are really compatible with organic solvents, and actually they are able to be stable even in harsh conditions. And also the preparations requires a really few amount, of price. It's a lower price, the preparations, and a really cheap protocol, what is involved for the production of MIPS. MIPS have been used in a wide variety of formats, like films, gels, nanostructured materials, and are applied to different fields in analytical. For example, they are used in catalysis, in control release, analytical separation, and in what we are putting the focus today in sensors as well. So for using molecular imprinted polymers in sensing, there are some disadvantages that should be conquered, to be resolved. So one of the first ones is that in general, they present a slow binding kinetics. For this, in the recent year, there are several strategies to make formats of MIPS that are able to facilitate the diffusion of the analyte to the cavities. And one of those examples is the synthesis of MIPS in the form of microspheres. Another disadvantage is the analysis of target without intrinsic optical properties. Well, this is common for any other sensing platform. When you want to analyze targets that don't have this possibility, the physicochemical property to be followed. So in the case that I'm going to show you, what we do is the synthesis of a fluorescently labeled analog for the implementation of competitive assays. Another disadvantage is it sometimes is difficult to couple the MIP to the transducer. So as a way of resolve this problem is the possibility of using fiber optic microarrays that are going to locate the microsphere sensors in there. And another disadvantage is also common in another sensing schemes is the difficulties that sometimes we have for doing multiplexing. So in the case, the easy way of obtaining the multiplexing possibility is encoding the microspheres that are going to be the imprinted polymers or control polymers or other kind of MIP microspheres, encoding them to identify inside the microarray. In this case, the antibiotic was the case study. So antibiotics are, as you know, are administered in therapeutic and as therapeutic and prophylactic agents in animals devoted for food stuff. And an uncontrollable use or disposal may give risk to the presence of antibiotic residues, creating challenges like in different fields like sanitary, analytical, technological, and environmental. The common problems in the clinical area is that they can promote allergic reactions, and also the possibility of getting superbugs for getting bacterial resistance. Also, in the case of the analytical analysis, I mean, in the case of some analysis that they can get false negatives, and for example, in microbiological assays. In the field of the technological, in the production of dairy products, like for example, they can inhibit the production of cheese if they are affecting the microbiota that is involved in this production, and also in the environmental field. There was a lack of a general procedure for the preparation of MIPS, I mean, of monodispersed MIP microspheres. So what we decided is to explore different composition by the help of experimental design in order to find out which was the best proportion. From all the possibilities that we had, these are the reagents that finally we used for obtaining these microspheres. The synthesis procedure follower was one of the most widely used, that is the precipitation polymerization procedure. In this approach, polymerization takes place in a large excess of the organic solvent, where the monomers and the template are soluble, but the resulting polymer is not. The synthesis was performed at 60 degrees during 24 hours, and after centrifugation, washing to remove the template molecule and vacuum drying, we obtained the particles. Several parameters were also optimized, not only the composition, but also the volume of the solvent, the type of the solvent, and also the amount of the cross-linkage that we used for the preparation of the microspheres. At the end, with the optimal composition, we were able to obtain really homogeneous MIP particles with a diameter of 3.2 micrometers that were able to specifically bind enrofloxacin, which was the antibiotic under study, and no other antibiotic. So it was a polymer. They were a microsphere really selected to this antibiotic. For the construction of the platform, of the sensing platform based in a competitive assay, another thing that we need, as I said before, is the analyte labeled with a fluorophore. So for the selection of this fluorescent moiety, there are some things that we need to take into account. For example, the fluorophore should have an absorption in the visible region. Should have also has a high luminescent quantum yield, chemical stability, and of course should be commercially available or easy in the synthesis. In this case, what we select is a label that it was body P, a body P label. Specifically it was body PTR cadaverine. This molecule that you have here was successfully synthesized, and we have a really good spectroscopic characteristic. For example, a really nice quantum yield, and the excitation and emission was in a good interval for be used in the sensing development. The sample plasmon was based in a PET-based microarray using fiber optical bundles. In this platform, the particles, in the case the MIPS, and the control particles that are the non-imprinted microspheres, were randomly loaded into the edges walls of an optical fiber bundle. This is an optical fiber bundle that is composed with thousands and thousands of optical fibers. And in each fiber, we do a well. So what we obtain in that way is a sensor, is a multiplexing sensor that is able to be monitoring what is on the top, that means the microsphere, by the use of a microscope. How we can distinguish, also how we can do the multiplexing? Well, to distinguish the different particles that we can load into the fibers, what we do is encoding them with different fluorophores at different concentrations. That means that in the case, for example, of the imprinted polymers, those microfields that are able to recognize the antibiotics, the color for identification is going to be coumarin 30. And in the case of the non-imprinted particles that are not going to recognize the antibiotic, are going to be encoded with ruthenium complex. With this encoding, we were able to distinguish by imaging those particles that are the MIP from those that are the non-imprinted MIP. This is a proof of concept because the idea is that we can use different imprinted particles for different analytes at the same time. After loading the fiber optic with the microspheres, what we do next is the competitive assay. So we first load both NIP and MIP particles into the wells. We incubate with the enrofloxacin cake with the fluorophore and with the analyte itself. They do a competition for the specific sites of the imprinted polymer. And finally, we wash out the excess of the reagents. Then we make the image acquisition, and this signal is proportional to the concentration of the analyte in the sample. The analytical characteristics that we're obtaining with these calibrations was also really nice, and the limit of detections using this approach was also really interesting. Another interesting point in this strategy is that with only 60 milligrams of those particles, we are able to perform more than 30,000 assays. The cross-reactivity that we obtained using this approach was also really nice because not other antibiotics that are not related with the enrofloxacin are not giving any signal in the system. Only we have a kind of good signal, good response or significant response coming from the ciprofloxacin, what is the metabolite of enrofloxacin, and what it makes is interesting in itself because that means that we are able to monitor not only the administration of the enrofloxacin, but also the metabolite that is produced once that the animal is treated with this antibiotic. The analysis of real samples was performed using blood coming from SIPs that had been treated with this enrofloxacin. And as you can see, the results were completely fully validated by a chromatographic method. And we can, with no doubt, say that the sensing platform was really useful and it was a real example of it. So thank you to everyone for attending to this conference, and I would like to acknowledge to all the people of my group that all these results cannot be done without every single one, and also to our collaborators and the funding that we got. Thank you very much.
Okay, we have not questions by the moment, but I encourage everybody to ask questions. I have a lot. I like a lot your seminar. It was a very nice seminar and very interesting also. And if you have questions, Maricruz, you can do it. But if no, I start because I have a lot.
Go ahead, please.
Okay, thank you. Well, the first question is in terms of the molecular recognition, the optical sensing systems that you included in the first slide, that you say that you can choose these optical sensing systems for different purposes. And my question is, if you want to detect a protein, because I always speak about, from the point of view of biochemistry, and I work with proteins, then if you were to detect, to sense a protein, which is the best system to detect, to use for these purposes? If you want to always to detect a protein, no a small molecule.
The sensing platform or the recognition element?
The recognition element.
Okay, so in the context of my presentation, I will say that probably the more reliable is using phase display, displaying fragment antibodies. Okay. That could be the best option.
But which is the size of the peptides that you recognize in these libraries? Because there are different phase display libraries, no?
Yeah.
Small and large peptides. Which is the one that you use usually?
Up to now, we are working with two commercial available libraries. And I know that from that company, we can get 3 different. So one is 12 amino acid peptides, which are displayed in the libraries, and the other is 7. That could be either cycled or not. Okay. So these are we know that there are other libraries, but these are the most common that you can work with.
And these mimotopes that you that you discovered, you analyze the existence of different mimotopes in these assays, these phase display assays. These mimotopes are, I understand that are linear mimotopes.
In the presentation I made, yes, but it's true that we have already get another mimotope for other, for other examples, other platforms, other analytes, okay, that they were cycled. So now, up to now, I can say that we have 50 linear, 50% cycles. This is the 2 types that we are working with. Okay.
Well, I have a question of how to use the term toxin. I usually use toxin meaning for a protein, a toxic protein, and you use toxin for other kind of compounds. Small compounds you are using always this. I don't know if you use indistinctly the name of toxin for small compounds, for big proteins. I don't know.
No, I was just focusing my speech in that mycotoxins that are the contaminants. That one of the case studies that I was talking about, they are toxins. They are natural toxins, small molecules that are metabolites produced by the fungi. And I know, of course, that there are other kind of toxins, that there are proteins that also are toxin, but in this speech, we were focusing our results that we obtained for small molecules of interest. And our work works essentially in the detection of those molecules that are interesting because they are contaminants. At the beginning, years ago, we were working with antibiotics, and the problematic that could happen, the prevalence of those residues. And now we are more focused in the detection of mycotoxins.
Okay. And also my last question, because the molecular imprinted polymers that you use, that you mentioned at the last part of the talk, how you can use this system also for a protein? You can use this system for a protein. You can design the cavity of in this kind of matrix for this.
Yes. At the beginning, it was a really challenge. There are not so many papers related to that topic because it was complicated. The proteins are flexible. You are preparing a recognition element that is not. So in the last year, and also in our case, we prepare some approaches for the detection of protein. What we are focusing is the detection of epitopes of that protein. It is possible to elucidate what epitopes of that protein could be used for with no doubt recognize that protein. And those small peptides is really easy. I mean, it's easy to imprint in a polymer and use it like a tag. Actually, not for sensing, but for purification of protein, we made that approach in that way. We prepare molecular imprinted polymers that are able to selectively bind flag tag proteins. So those proteins that are with the epitope flag were able to be purified nicely using those materials, those MIPs.
Okay, and only one more. You say that you can use different colors to detect in the MIP arrays.
Yes.
You can use different colors to detect the compounds.
It's a rational mixture of fluorophores.
How many? How many colors you can use? I mean, how many different colors you can use in this kind of assay?
Well, I think that there is something that is already commercially. I mean, Illumina and Luminex, all these platforms that are so common are based on those approaches. So basically, I think that now maybe they are in 500 possibilities. What they are using is a rational encoding with different concentration of 2 or 3 fluorophores. Those combinations allow you to have different identities for, I would say, probably 500 bits, I think now, maybe. Yeah.
Okay, thank you very much.
Oh, welcome.
I don't know if Maricruz has more questions.
In other responses.
Well, you know, it's a topic I know very well, but yeah, I would like to get your opinion about really the potential that peptides have, these peptides have for the implementation of assays for other targets. So in your opinion, and considering the experience that we have, as when Maite was asking about the use of linear or cyclic libraries. So what do you think? Which one is best from your point of view? And do you think that this technique can be open to the analysis of other targets?
So related with the cyclic or non-cyclic peptide libraries, I would say that the cyclic are more, it's probably more easy to get a really good mimotope, a peptide mimic of the target that you are aiming to, because the conformation that you are getting in the cyclic form, it's probably more easy to mimic the epitope. I mean, the complementary of the part of, for example, of the antibody. So, and actually, this is in the way that the upcoming commercially peptide libraries are going to. I mean, by time, the new libraries that are in the market are basically the kind of cyclic peptides, because probably it's more, they are good in the way of associate to antibodies or any other protein target. And in the case of if it is possible to use these libraries for getting mimotopes of any other analyte, yes, of course. I mean, in the case of small molecules, I think it's something that is possible. We have to take into account that the libraries contains billions of possibilities of different amino acid sequence in the peptide. So the possibility to find out a sequence that is going to resemble partially an analyte, I think is something that, I mean, there is no limit. I think that could be done with any analyte. And in the case of proteins, well, they could be probably mimic partially part of that protein. I mean, I was saying before with the epitopes, parts of the proteins can be mimetized by these mimotopes and be used for other purposes.
Yeah, because I was thinking that there is a possibility also to, if we compare it with the epitope imprinting approach, for instance, we could also try to do epitope selection by phage display. obviously, well, this is a random process and it's difficult to know exactly which part of the molecule is being, at the beginning is being identified, unless we imprint the epitope. We do a phase display of the epitope. I don't know if that would be possible, and then transfer that to the whole protein. So just selecting the peptide to do the phase display, and after that going to the full protein in order to see if you get rebinding, which is finally what we are doing with Imprinted.
Actually, something that it's done with phase display. I mean, trying to find out with a protein or autoantibody that need to be recognized. They put the library in there with the peptides, and it's just a question to see which complementary protein is linked, and find the best peptide that is able to bind these targets, these protein targets.
And what about phage display of recombinant antibody fragments? What is your opinion about that?
I think that it's a really interesting alternative to animal-derived antibodies that we know from the recent year that is an aim from the European Union, for example, in order to take out from the animal use for these purposes. It's true that the fragment antibodies sometimes are not working, depending on the condition, are not working as well as the natural ones. But for sure, this is a strategy that should be evolved to get best fragment antibodies for the substitution of the natural ones. It's in the way of using them, I think is easy. I mean, it's similar to the peptides library. It's true that is maybe for getting the best one, just 3 or 5 rounds is not something enough. If you are starting from a naive library, maybe for getting the best antibodies, you need to do a first round coming from a naive library with no biases. And then when you have those targets, those phages that are kind of able to bind in a good condition, then you need to do another library, in this case biased to your target, and in that you can increment the good characteristics toward the protein that you are aiming to recognize or this. So this is something that is already done, but it's more devoted for the clinical, for therapeutic. So what we probably need as analytical chemists is to get this technology with easy access for us for implementing assays, analytical platforms.
So you need a collaboration with somebody that is expert in the production.
Probably yes, because also this is like a close club that and companies that, for example, if you want to work with a library of those characteristics, what is actually difficult to produce these kind of libraries, it's a question of, it's a really expensive, it's more than 1,000 euros, for example, buying this kind of libraries.
Okay, any more questions? Somebody would like to ask something to Elena?
I think, Elena, that is right. I think that to use antibody fragments, I think that is the future, because even for if you want to label these antibodies, it's going to be easier than with long antibodies, polyclonal or monoclonal antibodies, and also the restrictions that we have in order to use the animal models to obtain this kind of antibodies. I think that we have to move to the recombinant products, and then I think that is the future for sure for this kind of detection system. Then I think that is something that you have to, and of course there are groups that can collaborate here in this faculty. Then I think that is going to be a future collaboration between different departments. I think that I think that is one of the purposes of this kind of seminars. I mean, to find connections and network between the different departments, and I think that is the idea.
Yes.
Okay. Then I don't know, there are no more questions, but I think that we have a clear idea of your work. And the only thing that I want to say is thank you, Elena, for your seminar. Thank you, Maricruz, for introducing her. I include in the chat the link of for the next seminar on March 17th. Agustin Molina is going to give the seminar. It's from the Organic Chemistry Department. And well, thank you everybody, to the audience for your attention, and see you for the next seminar. Okay, bye.
Thank you, Maite.
Thank you.